Hello, everybody, and I would like to welcome you to this webinar. Today, we're very privileged to have JoAnn Flynn giving this talk. I'll just introduce Dr. Flynn before she starts her presentation. Dr. Flynn is a distinguished professor in the Department of Microbiology and Molecular Genetics and the Center of Vaccine Research at the University of Pittsburgh School of Medicine. She obtained her Bachelor of Science in Biochemistry from the University of California at St. Davis and a PhD from the University of California at Berkeley in microbiology and immunology. And Dr. Flynn has been a distinguished professor and her research, distinguished professor since 2019, and her research focuses on TB immunology. And today she's going to talk to us about the path to sterilizing immunity against tuberculosis. JoAnn, it's a pleasure to have you, and please go ahead with the talk.
Thank you very much, Henry. I'm pleased to be here, and I appreciate the invitation and the chance to talk to people about some of our work. I think many of you know that tuberculosis is a bacterial infection transmitted by aerosols, in which case the bacteria are breathed into the airways and transit to the lung parenchyma. About 90% of those who are infected do not develop disease, which is called latent TB, which is a clinical definition. And that accounts for about 2 billion people in the world. And those people remain asymptomatic, sometimes for their entire lives, but sometimes they reactivate to active TB. A small proportion of people actually develop active TB within a year or two of being infected. And these people are symptomatic and can spread the infection and can succumb to the infection. When the bacteria enter the lung parenchyma, they form something called a granuloma. So the host response to the bacteria actually forms the granuloma, and it's a collection of immune cells that surround the bacteria and try to prevent it from spreading throughout the lung. So this remains a major health issue worldwide. So 10 million active cases every year, 1.5 million deaths per year. And I think it's important in the context of this seminar is to remember that most people who get active TB were actually vaccinated with BCG, the only approved vaccine for tuberculosis at birth, and yet they still get the disease. So we obviously need to do better, and that has been really a focus of our research for a number of years. So I'll be talking a lot today about a granuloma. So a granuloma is an immune microenvironment, and this is a sort of classic granuloma with the middle being necrotic, we call that caseum, surrounded by epithelioid macrophages, which are very differentiated macrophages, and then with a rim of lymphocytes and more macrophages. And the bacteria can live actually within the caseum as well as within macrophages. And it's a very interesting structure, so you can think of it as three-dimensional, and it can be small, less than a millimeter, up to, you know, maybe even a centimeter. So this is a source of bacterial growth, persistence, and killing, depending on how well the granuloma is doing. And in a lot of our data so far, we've demonstrated that these are dynamic structures, so they can change over time, and they can be quite independent from each other, even in the same host. So I work in non-human primate models of tuberculosis, and I do this because they really recapitulate the full spectrum of human infection and disease, as well as the pathology. So I'm going to talk today about work in two different models. So we have the Chinese cynomolgus macaques, which are really, if you infect them with a low dose, they actually present with a full range of human infection outcomes and pathology. So everything from what would be termed latent TB all the way to active TB and everything in between. So these are really great for studying sort of basic immunology, how does the host control infection, and reactivation TB studies, which I'm not going to talk about today. The other model that we use is the rhesus macaque, and it's more susceptible. Nearly all animals infected with a low dose will develop active TB. They also have human-like pathology, and because all of the unvaccinated animals will develop active TB, these are very good for vaccine studies, so I'll talk about them at the end today. So in order to understand immune protection in TB, you actually need to have some good models of protection. And so today I'm going to talk about 3 different ways we think about protection. So natural infection, which is in the way I'm going to talk about it today, protection at the granuloma level. I'm going to talk about concomitant immunity, which is protection against reinfection. And I'll end with a brief discussion of our work in the space of intravenous BCG. So we've developed over the last 20 years many tools and technologies for studying TB in non-human primates. For those of you who work in murine systems, non-human primates are as variable as humans. They are not inbred, and they can have a number of different outcomes, which makes them challenging to study, but also reflect the human situation. So we, in collaboration with Sarah Fortune's lab, developed barcoded MTB bacteria. So every bacterium has its own specific DNA barcode, so you can track infection. We infect with a very low dose, less than 20 bacteria delivered intrabronchially. We developed serial PET-CT imaging to track infection over time, and that's shown in the middle there. Sorry, I don't have a pointer in this format. We can do serial blood and bronchoalveolar lavage on our animals, and then we use the PET-CT imaging to do detailed PET-CT guided necropsies so that we have a map of where all of the disease is, all of the granulomas, which is what those arrows are pointing to. And we go in then and we match them to the PET-CT scan and obtain each individual granuloma for analysis, as well as any other pathologies and the lymph nodes, and as well as random lung. On these samples, each individual sample, we do quantitative microbiology, so we determine the actual bacterial burden in each granuloma. This chromosomal equivalence, which is the determination of how much DNA there is from MTB, and that's an indication of how many bacteria were ever there, as well as barcodes by sequencing. As I said, we have a PET-CT history for every granuloma we take out, so we know how inflamed it is, what the size is, where it is in the lung, as well as the timing. When did it arise? And that's important. We can do immunohistochemistry on the lesions. We do 25-color spectral flow cytometry, and then we obtain samples for our single-cell RNA sequencing, system serology, and innate assays. And I'm just going to really talk about the single-cell work today. So this is the first part is about natural infection, and this, I think, illustrates what I'm talking about. So this is an animal who has active TB. So this animal is 30 weeks post-infection. And on the top row of scans, so we scan this animal then 4 times over the course of 10 weeks, the black arrow is pointing to a granuloma, and this is in the right upper lobe. And over time, as you can see, that granuloma actually disappears so that it resolves by itself. At the same time, in the bottom, that the granuloma, that there are 2 granulomas shown. One grows quite a bit bigger and overtakes the other one and becomes a consolidation. These 2 things are happening simultaneously in a monkey with active TBs. So in the case of active TB, a monkey can resolve a granuloma at the same time that another one gets out of control. And we've actually validated this with Cliff Berry in his work in human TB PET-CT imaging, where he sees also dynamic changes even in the context of active TB. So granulomas are very heterogeneous, and they're heterogeneous in almost all ways. So this is shown sort of a typical granuloma in immunohistochemistry. You can see the B cells are in clusters, whereas the T cells really surround the outside of the granuloma, and with some on the inside as well. But there are a number of different cell types in the granulomas that we've determined by flow cytometry, and they can vary in their positioning and their numbers across granulomas, even in the same animal. We demonstrated previously that almost every granuloma begins just with one single bacterium, yet the trajectory of the granulomas changes over time. And so each granuloma we consider to be independent, although of course there is a host signature for every granuloma. In addition to cell composition, they also are quite heterogeneous in terms of bacterial burden. And so this graph shows a number of different animals. Each column is a monkey. Each dot is a granuloma. And this is shown in a log 10 axis, on the y-axis, and weeks post-infection on the x-axis. And you can see that early on, 4, 5, 6, 7 weeks post-infection, the actual granuloma bacterial burden is pretty high. And so even though the monkeys in general are not sick, it's all contained within the granulomas. But over time, the bacterial burden decreases so that by 10 weeks post-infection, actually, you're starting to see a lot more sterilization of granulomas, which is on the bottom of the y-axis. And you can see that this continues over time up to 24 weeks. So even though these animals, some of them have active TB, they actually can sterilize granulomas. But this doesn't really come into play until about 10 weeks post-infection. And what we think is happening there is that it takes a long time for the adaptive immune response to really gear up to be able to kill the bacteria in the granulomas. So putting all that together in a lot of data I haven't shown, if you think about this being a monkey and each apple being a granuloma, there are lots of different kinds of apples and granulomas. But it turns out that if you have one bad one, which is that ugly looking apple that has a bite out of it, if you have one bad one, actually that can lead to active TB. So you can do well in all of the granulomas and one of them goes bad, that can lead to dissemination and worse pathology and active TB. So we need to understand really what it is that makes a granuloma good or bad in terms of bacterial outcome. So how do we do that? So how do we determine which granulomas, which factors lead to resolving a granuloma or killing the bacteria versus a permissive granuloma that allows for MTB growth and dissemination? Whoops, I forgot how to do this. Okay, so we've taken a number of approaches to this, including, as I said, flow cytometry, but I'm just going to talk today about our single-cell RNA-seq work. And this is a collaboration with Alex Shalek's group, Sarah Fortune, Sam Behar, and the people who really did the initial work on this are Travis Hughes, who was a graduate student in Alex's lab and is now back in medical school, and Hannah Gideon, who's a research assistant professor in my group. So for this initial study, we had 4 cynomolgus macaques. We gave them a low-dose infection, and we followed them by serial PET-CT for just 10 weeks. So 10 weeks, remember, is that pivotal time point where the bacteria are starting to be killed in some of the lesions. We individually isolated the granulomas and determined bacterial burden and did chromosomal sequencing for CEQ. And then we, at the time of necropsy, we chose 26 granulomas just randomly from the 4 animals for single-cell RNA sequencing. And we did all of this in the BSL-3, so we use the SeqWell system that Alex Shalek has developed. So it turns out that when we looked at our data, although we randomly chose granulomas, when we went back to the PET-CT scan data, it turned out that they were a mix of original granulomas, those seen at 4 weeks, which is the earliest time you can reliably really see granulomas in the PET-CT scan, and that's shown in the on the left with the green arrows, and some that were late, so seen only at 10 weeks, so only the pre-necropsy scan. You can see that in the blue arrows on the right-hand side. And so it turns out that the granulomas arise at different time points, and we considered that this might actually result in some differences in the granulomas. So how did they arise? Well, dissemination. So we've proven previously with our barcoded strains that a granuloma can give rise to a new granuloma as a bacterium escapes from the granuloma, and we can track that by barcode. So that it could be that those that arrive at 10 weeks are due to dissemination, so they would form in the context of a different immune environment, but it's also possible that there are more slowly growing granulomas that we can't detect until 10 weeks. So when we now go and look at what the bacterial burden is in these granulomas, so on the left, shown in the different colors, are the randomly chosen granulomas from the 4 different macaques and the bacterial burden in log 10 on the y-axis. You can see that there's a variety of different bacterial burdens and some differences between the monkeys as well. And then if we look at time of granuloma establishment, early, which is 4 weeks, or late, which is 10 weeks, you can see that, in fact, there's a difference in bacterial burden in the early and late granulomas. And that's shown where it says CFU live. When we determine the chromosomal equivalence, which is how many bacteria have ever been in that granuloma, which means how much they grew, In fact, there's not a significant difference between the early and late granulomas, suggesting that the bacteria grew to approximately the same level. But however, when you look at the live bacteria versus the total bacteria, so CFU over CEQ, in fact, what you can see is that the late granulomas are much better at killing. So lower numbers here mean more killing. So that suggests that there's a difference between early and late granulomas. And just to demonstrate that we're not, that if we just did this straight as bacterial burden and we took all high burden lesions and all low burden lesions, you can see that they separate out at about the same, so that all of our low bacterial burden lesions are the ones that arrive late. So we decided to analyze them based on their time of establishment, early versus late. So when we do the single cell analysis, we see what the TB granuloma landscape looks like. So again, this is 26 granulomas from 4 animals, all sort of smushed together. And you can see there are a number of different cell types. So of course there's macrophages, so that's in the light green, neutrophils, a couple of dendritic cell subsets, some B cells in the purple, some stromal cells, fibroblasts and endothelial cells, plasma cells. And an interesting thing that we saw was the presence of mast cells. So you can see that's that big green colony up there. And we didn't expect to see mast cells in the granulomas, but it was they're actually a reasonably decent population. But the biggest population is the blue population, which separates out as T and NK cell population. So we call this the unified T-NK cluster. So when you just look at sort of a average of all the granulomas, and this is just to demonstrate early versus late detection on the top and high and low bacterial burden on the bottom. You can see they're essentially the same. So early detection granulomas have more macrophages and fewer T cells, and late detection granulomas have more T cells in the blue and fewer macrophages, with some differences as well in the other populations that I'll talk about. And just to demonstrate again that the late detection granulomas are the same as the low bacterial burden granulomas, you can see that in the pie charts here, whereas the early detection granulomas are the same as the high bacterial burden. So early granulomas have higher bacterial burden, late granulomas have lower bacterial burden. And this is due to killing, remember, not just bacterial growth. So what separates these types of lesions? And what you can see is that this is all of the subtypes. You can see that in the gray, which is a little hard to see, but the ones on the left in each column are the early granulomas, and the ones on the right are the late granulomas. So you can see that there's more T and NK cells in the late granulomas compared to the early granulomas. And then if you go across all the different subsets, what you see is that in fact, plasma cells and mast cells are higher in the early or low or high bacterial burden granulomas compared to the late or low bacterial granulomas. So this is an interesting thing that we've actually seen in other studies now, that plasma cells and mast cells correlate with higher bacterial burden. But just to keep this short today, I'm just going to talk about the T and NK cells. So we took this large T and NK cluster and we subclustered it. And it turns out that the subclusters that come out are not strictly CD4 or CD8, but instead they subcluster based on function. So we have 6 cytotoxic subclusters. We have a T1, T17 subcluster, a proliferating cell subcluster, Tregs, and a stem-like cell cluster as well. So how do these sort out in terms of early versus late granulomas? So we sort of highlighted the ones that are statistically significant, so that you have a cytotoxic subcluster, C4, which is higher in the late granulomas that are early, that have a lower bacterial burden. And you can see the stem-like cells, the regulatory T cells, the proliferating cells, and most prominently, the T1, T17 cluster. So these are cells that all correlate with lower bacterial burden or late detection granulomas. So we looked at this T1, T17 cluster, and it's a hybrid cluster, and it contains a lot of the features associated with perhaps TH1 star or XTH17 in terms of transcription factors. There is some interferon gamma and TNF, But interestingly, although there is ROR alpha and ROR gamma, in fact, we don't actually see any IL-17 expression at all in this cluster. So it's an interesting cluster, which we've just called T1T17. The reason we call it T1T17 instead of TH1TH17 is actually that it's composed of both CD4 and CD8 T cells. So when we looked at this, we noticed that there seemed to be some structure in this subcluster. So we subclustered that further into subpopulations, and we found 4. And one of them is quite small. Cluster 4 is a very small cluster, so I won't talk about that. But just to explain that cluster 1, the red cluster, is actually mostly CD4 cells. And in fact, they resemble a little bit of the stem-like cell clusters, and they are correlated with late detection or low bacterial burden granulomas. Cluster 2 is a cytotoxic cluster, expressing granzymes and perforins, and mostly CD8, but also has some CD4 in it, and it is also associated with lower bacterial burden. Interesting, cluster 3 is where most of the TNF and interferon gamma is, and it's both a CD4 and a CD8 cluster, and it does not correlate with bacterial burden. So the primary things that we always think about when we think about TB is interferon gamma and TNF are important. And I'm not saying they're not, they just haven't sorted out that way in the single cell RNA-seq data. So now if we think about just the original granulomas, just the first granulomas that arise in the infection, and we now can see that there's a small difference in bacterial burden at 10 weeks in those granulomas, and we look at the high and low bacterial burden just in the original granulomas, what correlates with bacterial burden? In other words, what correlates at the earliest stage of control? And it turns out that a number of the cytotoxic clusters, which contain both innate and adaptive T cells and NK cells, many of those cytotoxic clusters actually are associated with better control of infection in the earliest granulomas, as well as the T117. But in this case, now the interferon gamma TNF signature does correlate with better control in these early granulomas. So just to summarize the natural infection data, this is a very small piece of the work that we've done on this, is individual granulomas can be sterilized even in animals with active TB so that they act independently. However, looking at the early established granulomas versus the late established granulomas suggests that granulomas that arise in the context of a primed immune response, so arrives after the initial infection, have different characteristics and improved bacterial control. Cytotoxic functions are associated with better bacterial control, both in the late granulomas and in the early granulomas. And so I think that, I've always been interested in CD8 cells and in TB, and cytotoxic functions have really, I think, been difficult to assess, but also somewhat understudied in the world of TB. And I think that most of our data now really suggests a strong role for cytotoxic cells in control of TB. And not shown is that depletion of CD4 T cells or all of the CD8 cells in the first 6 weeks of MTB infection does lead to increased dissemination in disease, suggesting a role for both of these cell types, of course. Okay, so now moving on to my next protection model is concomitant immunity, which is protection against reinfection. So a study that we published in 2018 in collaboration with Sarah Fortune, we took cynomolgus macaques and we infected 8 of them with MTB, low-dose infection, tagged with a DNA barcode for library A, and we had naive animals. We let the infection go for 16 weeks, and then we challenged them with the same strain, but a different library Q tag, so we could track the second infection. And then we waited 4 weeks, and we analyzed the data. So the question was, does primary infection protect against reinfection? And in fact, it does. So this is published, so I'm just showing you 2 graphs. On the top, it is new granuloma formation attributable to library B. And this is confirmed by PET-CT. So the blue is the naive animals, and the red is those animals who have a primary MTB infection. And all we're showing you are the granulomas that are from library B. So you can see 5 out of the 8 animals actually had very low numbers of granulomas, and 3 had no new granulomas at all. So this is significantly different from the naive animals. when you take those granulomas that did form and you determine the bacterial burden, you can see that most of the granulomas that formed in in animals with primary infection were sterile. So these are, again, the library B granulomas. And that's compared to the naive animals who have an average of about 10 to the fourth bacteria. So this is really robust protection from reinfection. So we wanted to determine what might be responsible for protection against reinfection. This is a study performed by Shari Ganshua, who is a graduate student in my lab. In her first experiment, she showed that if you treat with drugs, if you treat the primary infection with drugs, actually you reduce the protection. So in other words, live bacteria give you better protection than drug-treated bacteria. But it does not eliminate completely the protection against reinfection. So you lose some protection, but you don't lose all of it. So in this study where we wanted to determine if CD4 T cells are necessary for the protection against reinfection, Shari did this experiment. So she infected cynomolgus macaques for 9 weeks with Mtb library A, and then she treated with isoniazid, rifampicin, pyrazinamide, and ethambutol, so good TB drug, for 4 to 5 months to really get rid of all of the primary infection. And the reason that we had to do that was because now we were going to deplete the animals of CD4 T cells. And if you have a primary infection and you deplete CD4 T cells, the primary infection will go out of control, so it'll make it really difficult to understand this experiment. So here what we had were primed animals, drug-treated, treated with either anti-CD4 antibody or IgG as a control. And we continued that CD4 depletion prior to and during the infection with Mtb library B. We also had 6 naive controls who only were infected with Mtb library B, no depletions or anything. So what happened here is sort of interesting. So CD4 depletion did reduce protection against reinfection, but had variable outcomes. So not all animals acted the same way, which when you work in monkeys generally happens. So on the left, you see the new granulomas formed that are due to library B based on our barcoding. And what you can see is that there are more granulomas in the anti-CD4 animals due to library B, but it's not statistically significant because there's a huge spread. So some animals look just like the IgG, and some animals look just like the naive animals. If you look at the total bacterial burden due to library B, you can see that, in fact, there is an increase in the anti-CD4 treated animals compared to IgG. And you can see that the protection is not as good when you treat with drugs. But in fact, it's somewhere in between the IgG and naive. So yes, there's a loss of protection due to CD4 depletion, but it doesn't make them as bad as the naive animals. And then just to look at dissemination, we look at how many barcodes we see in the lymph nodes, in the thoracic lymph nodes, which is a sign of dissemination. And you can see that there are more unique barcodes in the lymph nodes of the anti-CD4 treated animals. It's similar to the naive animals compared to the IgG. So you basically, anti-CD4 depletion actually results in a loss of control of dissemination. However, it's not a complete slam dunk, right? So if you look at the number of granulomas due to library B, you can see that there are 2 categories: animals who look just like the IgG monkeys and animals who look just like the naive. So some animals appear to be more CD4 dependent, or some granulomas appear to be more CD4 dependent. So if you look at the bacterial burden for the library B granulomas, you can see exactly what I just said. Some granulomas look just like the IgG monkeys, and some granulomas look just like the naive in terms of bacterial burden, suggesting that CD4 independent protection can occur. So we did single-cell RNA-seq on this on these granulomas, and so if you just start with looking at the IgG, or those with the primary infection, versus the naive, to see which sort of cell types actually correlate with lower bacterial burden, you can see that the IgG monkeys have more T and NK cells, and that's really the biggest difference. I mean, there are a few minor differences elsewhere compared to the naive animals. And that makes sense. These granulomas are, again, arising in a context of a primed immune environment. And the T and NK cell subset, so this looks like a different subsetting of the T-NK cluster, and that's because we really have moved on to a more robust sequencing of these granulomas. And this is work done by Josh Bromley and Alex Shalick's lab, taking a subset of the granulomas from our primed and naive animals. And you can see that there's a big difference between the granulomas that are from the primed animals, in other words, those with a primary infection, versus the naive. And a lot of the difference really is in the CD8 subsets. So there's a couple of different CD8 subsets that you can see, and they all have different cytotoxic molecules. But two of the subsets that are seen in the primed animals versus the naive animals are CD8 subsets that are either granzyme K high or granzyme H high. And it doesn't mean that they don't have other granzymes. That's just one of the things that distinguishes them. You can see that on the right-hand side that, in fact, they're much higher in the primed animals. In addition, the T regulatory cells are much higher in the primed animals as well. And in the naive animals, what you see is an increase, like you would expect, I think, in proliferating T and NK cells, because those granulomas are just getting started, as well as CD8s that are just granzyme B high, and then a CD4 population as well. So what happens when you deplete CD4 cells and look at what the cell types are in the granulomas? This is a little bit of a complicated graph. On the left-hand side are all granulomas that have lower bacterial burden. And on the right-hand side are all granulomas that have higher bacterial burden, regardless of which monkeys they came from. And what you can see is that the naive in gray are all on the high bacterial burden side. So those granulomas are not controlling the bacterial infection. And on the left-hand side, all of the IgG animals are there, so the lower bacterial burden. And those are the animals that are controlling, the granulomas that are controlling the infection. But the blue bars are the anti-CD4 treated animals. And you can see that some of them have the cell composition in their granulomas of naive, and some of the granulomas have a cell composition similar to IgG. So it's sort of interesting that they sort out that way. And when you deplete with CD4s, what you can see is that it really does change the granuloma environment. And so what we get is a mix of granulomas with features of naive or concomitant immunity when you do the subsetting on the T and NK clusters. So if you look at the anti-CD4 granulomas down on the lower right, what you can see is that we get an increase in certain CD8 populations similar to the IgG in some of the granulomas, but the other granulomas look just like the naive. And this is just sort of shown in a bar-coded sort of way. And so it really indicates, I think, that there's CD4 independence mechanisms at work, because all of the CD4s are very well depleted in all of these granulomas. And so what actually happens during CD4 depletion is that you, of course, lose your CD4 cells. So you lose your type 1, type 17 cells, you lose all of your helper cells, but you also, interestingly, lose those two cytotoxic subsets, granzyme H high and granzyme K high, that were so important in controlling in the primed granulomas. And so this suggests that CD4 T cells actually has a role in maintaining certain CD8 cytotoxic populations in the granuloma. And I think that's somewhat unexpected. So we're speculating how that might happen. And there is a subset of T helper cells, so CD4 cells, that produce IL-21. They do other things too. And Sam Behar previously has shown that IL-21 is important for MTB control in mice. And based on LCMV models, IL-21 is important for CD8 cytotoxic responses. So this suggests that depleting CD4 T cells doesn't just lose all the CD4 function, you start to lose the CD8 functions as well. So just to briefly reiterate the things that I talked about with concomitant immunity, and this is still very much a work in progress, is that granulomas that arise in a primed immune environment have increased cytotoxic functions and improved bacterial control. And again, that sort of harkens back to our natural infection data as well. Depletion of the CD4 T cells just prior to reinfection impairs the CD8 T cell response and results in variable outcomes, indicating there's some heterogeneity in CD4 dependent protection. And in progress, of course, we will do CD8 depletion in the context of concomitant immunity. And we want to compare what the protection, why you lose protection with and without drug treatment of the primary infection. So just in my last couple of minutes, I just want to talk about our work in the context of vaccination. So we've actually done a number of vaccine studies in our non-human primate models. And to be honest, most of them don't work. And so we see very little protection using adeno-based vaccines, using subunit-based vaccines, and using BCG itself. And so you really, to understand immunity, you really need a good model where you have a strong protective response. So I'm going to talk about our BCG IV work. So first, we asked the question whether or not BCG, which is the vaccine that's given to babies across the world, and it's delivered intradermally at 5 times 10 to the 5th. And so we did this in monkeys, and then 6 months later, we challenged them with MTB. And in cynomolgus macaques, you see about a 1 log decrease in bacterial burden on the left-hand side, which is significant, but you can see that there are monkeys with still a lot of bacteria. And in rhesus macaques, you also see about a 1 log reduction in bacterial burden, but again, that's not significant. So we wanted to know if we could do better. So we decided to ask the question of whether or not the route of administration of BCG, which is a live attenuated Mycobacterium bovis strain, could change the protection that we see in rhesus macaques. So this is work done in collaboration with Bob Cedar, Mario Roeder, and Trisha Dara at the NIH, as well as my postdoc, Joe Zeppa. So we had a group of naive animals. We had an intradermal group, so we call it intradermal low, which is the standard human dose of 5 times 10 to the 5th. We increased the dose by 100-fold, and we called that intradermal high, so 5 times 10 to the 7th. We had a group where we gave 5 times 10 to the 7th by aerosol, BCG aerosol, to the monkeys. We had a group where we combined intradermal and aerosol. And then we had a group where we gave 5 times 10 to the 7th intravenously to the animals. So the BCG vaccinations were done actually at the NIH, where Tricia did flow cytometry on the bronchoalveolar lavage for the next 4 months or so. Then they were transferred to us at Pittsburgh, and we challenged them with MTB 6 months after BCG. We followed them for 12 weeks using PET-CT and various other clinical measures. And then we necropsied the animals to see how much bacterial burden they had. So these are data from the flow cytometry on the airways, so using bronchoalveolar lavage. And so on the top are the CD4 T cells that are making any of these 4 cytokines in response to PPD, which is just a gemisch of mycobacterial antigens. And in the bright blue, you can see over the course of 16 weeks that those are the low-dose intradermal animals. And you can see an increase in CD4 T cells in the airways, modestly. When you increase the dose of BCG to 5 times 10 to the 7th intradermal, you see an increase in CD4 T cells that is slightly above that of the lower dose intradermal. If you go over to the right side of the graph, the 2 green groups, those are the aerosol groups. And what you can see again is that you do see an increase in CD4 T cells when the BCG is given by aerosol in both of the green and the aerosol ID groups. But if you look at the red ones, those are the intravenous groups. And you can see that, in fact, we get about 100-fold more CD4 T cells responsive to mycobacterial antigens in the airways of the animals compared to the low-dose ID. And if you go down to the bottom graph, those are the CD8 cells. You can see there's actually very little increase in CD8 T cells in any of the groups, including the aerosol group, except for in the IV group, where again, we're getting about 100-fold more CD8 T cells that are making cytokines in response to mycobacterial antigens. So this is a massive increase in T cells in the airways of these animals following intravenous BCG. Just to cut to the chase, there's a lot of other data, but it's all published. These are on the left-hand side of the graph of the slide are our final 3-dimensional PET-CT scans of all the animals. And you can see that the unvaccinated group has a lot of TB, so anything red and orange is TB. The ID low, there you can certainly see a lot of disease. It's hard to see in the 3D graphs how much disease there really is. The ID high, a couple of those monkeys look pretty good, actually. The two aerosol groups have a lot of disease. In the IV group, we can see actually that just the far animal on the right actually has some disease in the lungs. And when you quantify that total fluorodeoxyglucose, which is the PET probe we use, so it's basically how much inflammation are you seeing in the lungs, you can see that in fact 9 out of 10 of the IV vaccinated animals had no inflammation in the lungs at all. And a couple of the ID high animals look pretty good, but the aerosol group looked very similar to the low dose ID group. And when you further analyze the data from these animals, if you look on the left-hand side, this is the number of granulomas that we found in the lungs at necropsies in each animal. And you can see that, that in the naive animals, there's a pretty decent spread, and it's similar to that you can see with the ID low dose. The only significant difference was found in the IV group. And so where 6 out of 10 of the animals had no granulomas in their lungs at all, and a small number in the other 4 animals. And then we calculate the total thoracic bacterial burden by plating and counting the granulomas and all other pathologies, plus the lymph nodes from the lung, to get a total thoracic bacterial burden. And what you can see is that ID low gives you about a 10-fold decrease, as we saw previously in rhesus macaques. There's not much difference with when you increase the dose intradermally by 100-fold. And that the 2 aerosol groups look essentially the same as the intradermal group, so no protection. However, and I think most remarkable is that 6 out of the 10 IV vaccinated animals had 0 bacteria in their entire body. This is true sterilizing immunity, whereas 3 of the animals had less than 50 bacteria, all contained within a single granuloma, and 1 animal was unprotected. So this is 90% protection. So what's going on here? How come this works so well? This is a little bit of a complicated graph, but it involves injecting the monkeys with CD45 antibody, anti-CD45 antibody IV just prior to necropsy. And then these animals were just 4 weeks post-BCG, so no TB, and determining which, whether or not they have T-resident memory cells. So this is a little bit complicated, and I wish I had a pointer, but on the left-hand side is bronchoalveolar lavage cells. They should all be CD45 IV negative, which means that they are resident. If they're CD45 IV positive, that means they're in the blood. And so if you look at the high-dose ID animals, you can see about across the lung lobe, somewhere between 10 to 28% are in the left-hand upper quadrant, which is the T-resident cells. You can see the aerosol is about the same, maybe 30% of the cells are T-resident. But in the IV group, which is the second row down, you can see that fully 80 to 85% of the cells are T-resident memory cells. And you can also see that there's a lot of BCG in all of the lung lobes at this early time point. If you do endobronchial, so you actually deliver the bacteria right to the airways, which Frank Varek has shown is actually protective against TB as well, where you deliver the bacteria, which is in the left lobes, you actually get very high levels of T-resident cells. And so, in fact, we think that IV, because it spreads the bacteria through the lung lobes, as well as primes cells in the spleen and all the lymph nodes, in fact, you're getting better T-resident cells. And we think that that may be actually important in control of the infection. And then just one final data slide. We've actually now down-dosed BCG IV across about a 500-fold range of bacteria, just to determine whether or not you had to have 5 times 10 to the 7th live bacteria IV, because that's really a lot of bacteria. And in fact, it turns out that across from 5 times 10 to the 7th down to less than 10 to the 5th, in fact, we can get sterilized monkeys, looking at the total thoracic CFU on the right-hand side, you can get sterilized monkeys at all of these dose ranges. Although you, of course, lose control as the dosing goes down. But I think it's remarkable that you can still get sterilized. Rhesus macaques, which are so susceptible to TB, you can still get sterilization even when you dose down to 10 to the 5th BCG IV. So just to summarize, we have robust protection against infection, and it really is against infection because some of these monkeys never even have a T-cell response to MTB-specific antigen, using BCG by the IV route. with complete protection against infection in 60% of the macaques and 90% overall, with a 100,000-fold reduction compared to low-dose ID. Interestingly, we did not see improved protection with aerosolized BCG, but we do see protection across a 500-fold range of BCG doses. We have large numbers of CD4 and CD8 T cells, and as well as T resident cells in the lung. We have initiated studies on depleting both CD4 or CD8 T cells prior to challenge, and in fact, it turns out that both of those cell types seem to be important in protection in data that I don't have time to show today. We've also depleted B cells at the beginning of the vaccination period to determine if antibodies or B cells are important, and those are in progress now. So just the take-home messages from today is cytotoxic functions correlate with bacterial control, and I think really should be paid more attention to in the context of TB immunity. And that granulomas that arise in the context of an immune response are much better at controlling infection. And this is supported by data from reinfection and natural control of infection, but also from BCG IV for those few granulomas that do form. CD4 T cells are important in concomitant immunity, but may also be regulating the CD8 T cell memory. We think it's possible to protect not only against TB disease, but also against infection in a very susceptible rhesus macaque model, with BCG IV driving high numbers of CD4 and CD8 T cells in the airways and T resident cells in the lung. And I think, I've been in this field a long time, and I used to think that it wasn't possible to get a really effective vaccine against TB, but I think it is possible, and I think that we're in exciting times now. So just to give credit to the people who've done the work, Hannah has done all of the single cell data that I showed you, and Kaylin has worked on CD8 cells. Pauline is our fantastic statistician and PET-CT analyst, and Jo has done a lot of the BCG IV work. And this is Ling, who has worked closely with me, as well as Chuck. And we're always looking for good graduate students and postdocs, so feel free to email me if you're interested. And these are my collaborators I talked about. Sarah, of course, is a fantastic collaborator. Mario, Tricia, and Bob. Alex's group, in particular, I showed data from Travis and Josh Bromley, but Sarah Nyquist has worked on our CD8 studies as well. Brian Bryson's a fantastic collaborator. And this is really funding from the NIH, as well as the Bill and Melinda Gates Foundation. And I appreciate your time, and I'm happy to take questions.
Thank you, JoAnn, for a fantastic talk. I do have some questions from the chat. So the first one is the CD4 depletion experiments. I mean, there's a question saying, how does this data correlate with CD4 depletion in individuals infected with HIV and TB co-infection, for example? Can you draw some similarities from the granuloma work?
So CD4 depletion due to HIV is what you mean, right?
That's correct.
Yeah. So Ling Lin and also Deepak Kaushal, who's separately done work on SIV and TB, as well as Chuck Skanga. It's actually interesting. If you just do CD4 depletion in the context of, for example, let's say latent TB versus SIV infection in the context of latent TB, you don't get the same answers. And so CD4 depletion does lead to some reactivation, but SIV leads to a lot of reactivation. And so suggesting that SIV is not just CD4, the results of SIV are not just CD4, suggesting again that HIV does a lot of other things to the immune system besides CD4. But in fact, you can sort of see the same thing, that not all granulomas are CD4 dependent. And so it's hard. I think we'll really have to look at the effects of CD4 depletion and HIV on all of the functions of CD8, rather than just looking at CD8 cells and whether or not they make interferon gamma or TNF, which is really what most people have done so far.
Yeah, okay, that's interesting. I mean, there's a lot of interest in the IV BCG experiments that you have done. The first question was, do you have an idea how long the protection is from IV BCG when you compare it to the standard intradermal?
We do not. We did what we felt was a pretty long experiment, waiting 6 months post vaccination. It's an experiment we'd really love to do. The problem is we have so many experiments with Bob and Mario that we want to do, and they're all long and expensive. But I think it's very important to carry these monkeys out for a year or 18 months to determine how long the protection really lasts. So we really don't know. I can tell you that, interestingly, BCG doesn't really hang around very long in the monkeys. Mm-hmm. So yes, at 4 weeks post-infection, post-BCG vaccination, you can find the BCG. But if you start with 5 times 10 to the 7 and you look for the BCG everywhere in those monkeys' bodies, you end up with somewhere in the range of 1,000 to 2,000 bacteria at 4 weeks. So I think, and we don't see it really at 6 months. So the BCG is gone relatively quickly in monkeys. And I don't know, Henry, if anybody knows really how long BCG hangs around in people. In mice, it hangs around forever. Yeah, I know. I mean, I've asked Barry about this, Barry Bloom, but I don't think we have a good sense of how long it hangs around in your standard, everyday, immunocompetent person. So, but the BCG doesn't hang around. So I think the question of how long the protection lasts is really important.
Okay. But from these experiments, the IV BCG experiments, do you think protection is both against latent TB and active disease?
In the experiments that we've done, all we know is that it protects against infection. So we don't know what effects it would have on an established infection, either active TB or latent TB. I worry a little that if in the context of active TB, in fact, there's a lot of inflammation induced by IV BCG, and you might make it worse. In the context of latent TB, I don't hazard a guess. I could go either way on that. And I think it'd be a really interesting experiment. It's just a super, super expensive experiment to do.
Right. Okay. So I'm just looking down the translational pathway. Do you envisage IV being a route for BCG vaccination in the near future?
Well, the near future, is that next week or is that in 3 years? No, I think we have a lot of work to do. And, Bob Cedar has done intravenous vaccination with sporozoites in the context of malaria in kids in Africa. And it is possible, I think, as a route. But sporozoites are irradiated, they're not alive, and BCG is a live vaccine. So we have a number of different thoughts about that. Obviously, you first want to determine whether or not the results are reproducible in humans in terms just of the immune responses, right? But there's also ways to modify BCG, and we have a project with Dirk Schnappinger and Eric Rubin and Sabina Ehrt and Sarah Fortune, where we're trying to make a BCG that kills itself. In other words, a suicide BCG. So you don't have to worry about that. But there's also really nice data from Chuck Skanga at Pittsburgh showing that BCG IV actually protects monkeys who have SIV. And in that context, he has, so the monkeys have SIV, he gives them BCG IV. 3 weeks later, he treats the BCG with drugs. And none of the monkeys get sick, and 90% of them are completely protected, just like the non-SIV monkeys are. So I think that suggests that none of the monkeys got sick. Maybe it'll be okay, but again, it's a long road to putting live BCG in somebody's arm.
Sure. And in terms of the safety profile of IV BCG compared to intradermal from the macaque work?
By single cell. So some of that was done in the BAL and that was published in the paper. But since then, we are now interrogating the lung tissue, the BAL, and any granulomas that do show up in the context of vaccination by single-cell RNA-seq. We just don't have those data yet. But I think that's going to be really important. And how does that compare to concomitant immunity, and how does that compare to natural protection?
Okay. And while you're on the issue of concomitant immunity, there was a question that, do you think treating individuals with anti-tuberculosis chemotherapy does increase their risk of reinfection and development of disease afterwards.
You mean people with LTBI?
Yes.
Presumably, right? It's a really great question, and I think actually our data suggest at least that not treating LTBI might provide more protection against reinfection. But again, we haven't done that, that long-term experiment that it would take to really answer that. But I mean, it does suggest, just like Leishmania, that having a few bugs around actually provides the ability to prevent reinfection. And I don't know, I mean, most of the world doesn't treat LTBI, right? I mean, it's just us in the States. But I that. But of course, in the context of active TB, that's not a question you treat, right?
Yeah. Okay. I have another question on IV BCG. I think you did mention that IV BCG vaccination was associated with low levels of inflammation in the lung. Is that correct?
Yes.
In terms of actual tissue damage, probably looking at was there any significant tissue damage, or did any of those animals?
No. So what you see, if you actually necropsy the monkeys at 4 weeks post BCG IV, you actually see microgranulomas,
Right.
and so they're not caseous; they're just little collections. You can't even really see them very well by eye, but you can see some, slight inflammation in the lung. That goes away, and so that we don't see any long-term. I mean, our pathologist has really carefully looked at all the monkeys.
Yep.
We don't see long-term tissue damage due to VIB. Again, we're not doing any tests on their lungs to determine if there's any effects, but they seem fine. The monkeys actually tolerate it extremely well. So,
okay. I guess one of the last questions I have here is the role of T and NK clusters in control of TB. I mean, that's quite fascinating. So how are you taking this work forward? And do you think it will give us any direction in terms of looking for vaccines for TB or anything like that?
Right. So I think that nobody is surprised that T cells matter, right? But I think one of the interesting things is that the cytotoxic subunits functions are really critical. And we never look, right? We never really look at cytotoxic and determine which cytotoxic functions are really important. So that's a road we're going down now, is trying to determine that. And then whether or not there's ways to induce those cytotoxic functions in the context of the vaccine. Because there are things that can induce cytotoxicity that are used in the cancer field and other fields. And so, in fact, I think that there are ways to start thinking about this that really incorporate the full range of functions that CD8 T cells can do, as well as not forgetting about the CD4 T cells as well, and particularly that T1 T17 subcluster, which is the most strongly associated, certainly, with control. Yep. So I think that it just opens up the world a little bit of that the T cells are not just CD4 T cells doing this and CD8 T cells doing this. It's really a range of different functions that correlate with outcome, and that we need to think a little bit more broadly than just saying, can we make some interferon gamma, which clearly has not worked so far, right? In our vaccine. No. Yeah.
So. Okay. JoAnn, this has been a really fascinating talk. I've exhausted the questions that I had jotted down. So there's a lot of chat. Thanking you for a wonderful talk, and I'm sure the audience have enjoyed it as much as I have. So once again, I'd like to thank everybody for joining this webinar, and special thanks to you, JoAnn, for taking the time to give us a fantastic talk.
Thank you so
looking forward to meeting you again next time. Thank you.
Hopefully in Malawi.
Yes, indeed. Thank you.
Thank you.
Bye.